Hi, my name is Daniel Pace-Farr. I'm a sophomore at ASU, currently in CHM 394, computational chemistry, and my final project presentation is about investigating the efficacy of nevirapine, which is a non-nucleoside reverse transcriptase inhibitor in HIV-1 drug therapy in regards to single point mutation in reverse transcriptase. So the general idea of this project was to perform computation at the HF level to analyze the energies of the binding interaction of HIV-1 reverse transcriptase to the non-nucleoside reverse transcriptase inhibitor, nevirapine, and the single point, and two complexes of reverse transcriptase, the wild type and the single point mutant tyrosine 181 cysteine. Crystal structures of these complexes were obtained through the protein data bank, and these calculations were performed to sort of confirm what we already know about first generation NNRTIs and how they can be improved, how they are already being improved in the second generation of NNRTIs, and the results compared with published material in this area. So non-nucleoside reverse transcriptase inhibitors are one type of drug treatment for HIV-1 to stop its replication at the reverse transcriptase step. So this is when HIV-1 single-stranded RNA is released into a host cell along with viral enzymes such as reverse transcriptase. Reverse transcriptase converts that ssRNA into dsDNA, which is then integrated into a host genome. Nevirapine is a non-competitive inhibitor, non-competitive allosteric inhibitor for HIV-1 reverse transcriptase. And that allosteric inhibition sort of contributes to single point mutations being having such drastic effects on its binding interaction in regards to nevirapine in the mutant complex. So computationally analyzing this loss of efficacy can confirm what we already know in regards to the second generation and first generation NNRTIs. So here on the left, you can see the structure of wild type HIV-1 reverse transcriptase in complex with nevirapine in red, and the tyrosine 181 residue is also indicated in red. You can sort of see it just behind there. And to the right, you can see the molecular structure of nevirapine, and actually the ring structure to the right side of that molecule is the part that will be interacting with the 181 residue. So in terms of theory, the restricted Hartree-Fock method was utilized to compute the various wave functions for each atom analyzed in the protein complexes, both wild and mutant types, and unbound and bound to nevirapine. And so in general, this is computationally expensive to perform such a calculation with a protein complex as large as reverse transcriptase. And to that effect, the minimal basis set STO3G was utilized to perform sort of just a rough calculation of this interaction, and the universal force field was calculated for each atom. And so the theory is that by calculating the total energy for each protein complex, the wild type and the mutant, with nevirapine bound and unbound at the binding site, the energy of binding can be determined. In terms of the actual procedure, the respective protein complexes were imported into Avogadro from the protein data bank. Those were the crystallized structures. Nevirapine, tyrosine, and cysteine were built in Avogadro for reference. Then Avogadro was utilized to calculate Z matrices of the reverse transcriptase complexes, wild and mutant, bound and unbound. These were inputted into Gaussian 16 using ASU's Agave computing cluster to run the computations and perform the relevant universal force field calculations. The total energies were recorded in log files, and then VMD was used to model and render the drug enzyme interactions that were relevant to this presentation. So we can break this down into 4 general steps. First, we prepare the protein complexes using the appropriate theory. Then we prepare the nevirapine and amino acid molecules for reference using the appropriate theory. We compute the respective energies using Gaussian of each protein complex, bound and unbound, with the target drug. And then we analyze the total energies obtained using Gaussian. So as you can see, the wild type results at the top and the mutant results at the bottom. The wild type drug enzyme interaction is energetically favored, while the mutant type drug enzyme interaction is energetically unfavored. And you can see to the left, overlaid over each other, 2 residues, and the nevirapine molecule in the wild and mutant types. To the left, you have the 181 residues, and to the right, you have the 100 residues, which are both leucine. So to the left on the bottom, you have tyrosine, and to the left on the top, you have cysteine. And you can see that in the mutant, which are in darker colors, the 100 residue of leucine is brought much closer to the nevirapine molecule, which could contribute to some sort of repulsive interaction. And of course, the side chain of cysteine is going to contribute to some sort of repulsive interaction as compared to the tyrosine side chain. So here you can see the relative positions of the 181 residue and nevirapine modeled. The side chain of cysteine, the sulfur atom is going to have a repulsive van der Waals interaction with the ring structure in the nevirapine molecule, and that is in contrast to the tyrosine molecule at the 181 residue, which is going to have an energetically favored interaction with the nevirapine molecule. So there are sort of 2 strong repulsive forces between nevirapine and nearby residues in the mutant reverse transcriptase complex, resulting from that single point mutation at 181 from tyrosine to cysteine. There's a partial repulsive interaction with leucine at the 100 residue, which is brought closer to the binding site due to the mutated conformation of the complex. On top of that, the sulfur atom in the side chain of the mutated cysteine residue contributes to a van der Waals repulsion with nevirapine, which actually contributes to that repulsion in a much larger way than the leucine 100 residue. Combining these 2 factors together explains the massive change in the energetics of the binding interaction of nevirapine to reverse transcriptase. And this magnitude is borne out by published data, which suggests that nevirapine is 113-fold less likely to bind with the tyrosine 181 cysteine mutant complex than the wild type reverse transcriptase complex. So we can draw a few conclusions from this, which are supported by published research in this area. Because the nevirapine reverse transcriptase interaction does not involve hydrogen bonding, it's a weak interaction to begin with, and it's also an allosteric, nevirapine is also an allosteric inhibitor of reverse transcriptase. So combining both of these problems, we can see that slight changes in the conformation of the protein close to the binding site can have drastic effects on the binding energy. Just bringing the leucine 100 residue closer to the nevirapine binding site by a few angstroms was enough to reduce its enough to introduce enough repulsion to weaken that interaction in a significant way. And so research has suggested that strong hydrogen bonding should be a key part of second generation NNRTIs to strengthen this interaction at the binding site, and indeed it is. And as for things that the simulation and computation that was performed did not take into account the presence of solvent. So some might question the accuracy of the energetic calculation in this reaction. However, we can say that because the drug nevirapine is neutrally charged, it most likely would not have a long-range interaction with charged solvent bound to the surface of the protein once it is bound inside the hydrophobic pocket at the binding site. Furthermore, the simulation computation does not take into account the kinetics of the interaction between nevirapine and reverse transcriptase, only the energetics. Therefore, there's not much we can say about the binding affinity of nevirapine to reverse transcriptase. However, we can say that in the mutant case, the interaction of nevirapine and reverse transcriptase is energetically unfavorable, whereas in the wild type, it is energetically favorable. That concludes my final project presentation for computational chemistry, CHM 394. Thank you for listening to my presentation. And here are my works cited.